Chor, who's going to give a talk on phylogenetic reconstructions based on RNA secondary structure.
How do I?
Okay, that's forward, and that's just forward, and that's a pointer, and that's back.
Good. Okay, so thank you very much. It's a pleasure to be here, especially when the weather is so great. No, I mean it. Okay, this is joint work with Moran Kabili, Asaf Meirovich, and Metzada Pasmanik from Tel Aviv University. And the basic idea is something which is not the first time that it occurs, is what are we basing our phylogenetic trees on? So I guess if this was, 200 years ago or something, then there was no other source rather than looking at all sorts of bones and or maybe, maybe wings, but basically morphology. Just because we didn't have any access to any other type of information. Once sequencing technology, once we understood, what life is composed of, DNA or amino acids, and we were able to sequence things, then, so I guess mid-60s or something, people started to have phylogenies which were based on either genes or DNA or proteins, homologous genes or proteins. But in any case, these are fairly short sequences of either type. And one may ask, maybe there are some other sources which could carry other sort of information, and which could maybe be of some interest. Okay, so one thing which, again, we have access to only starting in 2002, more or less, is basing phylogenies on whole genomes, just because there were no sequences of whole genomes before that. So initially, fairly short sequences, but now fairly short genomes, but now we also have access to large eukaryotic genomes. And when you try to look at things based on whole genomes, you get a picture which is not totally identical to any, usually is not identical to any given gene. So there were not too many, but let's say I think about 10 or 20 works along these lines using whole genomes. So our question was, could we use more biological sources to base phylogeny on? And basically, the work on whole genomes and the work I'll mention now and maybe another one, basically they have the same paradigm, which is basically metric induced. So you have objects, so whatever these objects are, whole genomes or some networks or whatever, or RNA secondary structure, which is what I'll talk about now. And then you have either to devise or to use some given good metric between pairs of objects in this domain. Now, so you have to first find a metric, and second, find a reasonably good algorithm to compute or to approximate that metric, because just having a metric, abstract metric, is not good enough. We want to compute things. Now, once you have an algorithm, compute those distances, and then, using, I wrote here neighbor joining, but I think even in this meeting, we'll have, 3 or 4 other proposals for building trees from distances. So I don't care. You could use any other one. So use any method for constructing trees from distances. And you get a tree, then it's also a good idea to do some sanity check. You get a tree, better not to put, man together with some bacteria and, and so on. So some sanity check that what you get is more or less reasonable. I don't think one could expect, if you use this approach, to get trees, which totally agree with the common taxonomy. But even if you build a phylogeny based on single genes, you don't get things which totally agree with common taxonomy. So I think it's good to have some diversions from the common agreement. It's not good to have, very wild things. So man together with bacteria is probably a bad idea. Okay, so what we so this approach was already applied fairly successfully. I'm just listing here two works that I was involved in, but there are a number of others. So for a whole building phylogenies based on whole genomes or proteomes, this was work which was in RICOM in 2005, and some other which was based on metabolic network was in RICOM in 2002, but there were other works as well. So that's something that one has to, well, I mean, it's obvious, but still I mention it. When you do something like that, the distance obviously has to fit whatever domain you're using. Distances for whole genomes will not work for distances for, RNA secondary structures. These are just two, these are domains which are very different from one another. So really the main, I don't know if the main, but one of the major things in this approach is to, for every such domain, to devise appropriate metric and to devise an algorithm which will work for it. Okay, so the question we raise here is, could we have phylogenetic reconstruction which is based on RNA secondary structure? So as probably most of you know, RNAs are, okay, there are messenger RNA, but RNAs do very many different things in the body. Recently we know about, not so recently, 5 years or so, about the mirs, how they are used, how they're used to control activity of genes. But there are many other catalytic, RNAs which have some catalytic activity. And I think these are, if you read Nature or Science, we discover every few weeks you discover another class of RNAs which have some important role rather than just moving information. And it's quite evident that the structure of so RNA doesn't have as complex structure as proteins, but still it has three-dimensional structure, and it's quite clear that, which is quite well approximated with this secondary structure. It's quite clear that this secondary structure is very important for the function of RNA. So maybe if we base phylogenies based on this secondary structure, maybe we get some information, which I'm not sure if it describes the whole evolution of the species, but at least it describes some interesting evolution of this RNA, which might be of interest by itself. So that was our goal. Okay, and so this is sort of I'll talk about it later, but yes, we did manage to do this thing, and we find things which are reasonable. I don't want to claim that they are the truth, but they're reasonable. For example, this is some tree based on 16S ribosomal RNA for 91 species. So at least you could see bacteria, eukaryotes, and archaea are very nicely split, are very nicely separated. So man is not together with bacteria, and also these colors indicate some families of bacteria. So they were also coded quite nicely by this approach. So there is, so as I said, we did have some partial success, or some success here. And what I want to describe is how we went about getting it. Okay, so what we did, you could go about it in different ways. We decided RNA algorithms which align RNA or compare RNA and so on, they're usually not, they're quite intricate, and the secondary structure of RNA. So we decided not to reinvent the wheel, but rather look in the literature and find something which existed and which to us seemed reasonable. And what we decided to take is that there are more alignment algorithms which are based on similarity, and they give you some score. So we decided to take something like that, particular algorithm like that, then transform similarity to distance. It's not totally, you have to work a little bit, but it's not totally out of the question. And then once we have this, because we want to use neighbor joining or something like that, so we need something based on distances. Once we have that, we want to find the appropriate RNA databases to get molecules and structures. That's not that easy, turns out, because initially we thought, okay, there is zillions of molecules, so we could take homolog molecules and just use some algorithm which folds them to the secondary structure. It turns out that's highly unreliable. When you do that, you have very similar molecules which the algorithm folds to totally different structures. I don't want to give examples here, but we have some examples. So instead, we decided to stick to databases where we have curated structures. Somebody looked at it, not just the algorithm, but somebody looked at it. It's really, I wasn't aware of, I'm not really an RNA person, I wasn't aware of how important this is. So it's really important to have something, if you want to have something reliable, not to trust the algorithms, but to use something which some expert looked at. Maybe that's a challenge for the algorithms, but that's not what we are going at. And then once we have that, we have reliable molecules, reasonably reliable molecules, and this distance algorithm, we just compute distances, run NJ, and then look at the outcome and see if it makes sense or doesn't make sense. One more thing which we did was comparing the outcome of trees from structure to the outcome of trees from from the same sequences, just the sequences themselves, that's easily available, and to see which one seems to be more reasonable or less reasonable. Because these are obviously two very close sources, the sequence and the structure stemming from it. Okay, the algorithm we used is called RS match. It's by four authors, appeared in bioinformatics in 2005. I will not go through all the details because it's not that trivial. Probably I need 15 minutes to describe just this algorithm. It uses what they call the dot and bracket representation. Brackets correspond to stems which are matched, which we got two sides which are matched, and the dots correspond to loops which are just single nucleotides which are not matched. The algorithm breaks the structure. Somehow it sorts those nucleotides and breaks it into substructures. And when it tries to compare two, it tries to compare those substructures. I didn't explain it too well, but you could go to the original algorithm. So RS match, also one nice thing, it employs both the structure, so it has access, its input is sequences in this dot bracket formulation, so it's secondary structure sequences, secondary structure RNAs, but also it has the sequence, so it also cares about whether the nucleotides are the same or not the same, and so on. So it uses both. And the last nice feature about it's a dynamic programming algorithm, probably every alignment algorithm, or most alignment algorithms, but it's also efficient. So it's just order of nm. If n is one sequence length and m is the other, it's just order of nm. What's that, some bug? Okay, so we took that algorithm and then we wanted to transform it from similarity to distance. Also, there were some small things we had to adjust in the algorithm itself, which I won't go into. But basically, the main thing was to change the distance, the similarity metric to distance metric. Obviously, that something like that is quite arbitrary. You could have many choices. Just like in the original algorithm, the choice of the exact similarity metric is also probably a bit arbitrary. So anyway, what we wanted, so lower similarity originally should imply higher distance. That's clear. We try to preserve ratios between mismatch values. So if there is some mismatch value, if there was high, if there was a high similarity, we try to have a low distance, and also to preserve ratios between different pairs, and also to have the usual requirements. So triangle inequality, symmetry, non-negativity, and of course self-distance should be 0. Okay, these are the tables. They are not that interesting. One thing you could see is that the distances between pairs. So we've got pairs and we've got single nucleotides. So the pairs corresponds to the stems, and the single ones correspond to the loops, which are sort of free. So the free ones, the mismatches got a higher penalty, higher distances, because somehow it seems that that's something that is mentioned in the original paper as well. It seems that the free ones have more somehow. They're the ones who do most of the catalytic work, so you penalize them more if they're not the same. Okay, gap cost was 3 per nucleotide. So if it was just singles, it was 3. If it was pairs, it was 6. And this means that mismatch is less than twice the gap cost, and that guarantees triangle inequality. So that wasn't too hard to guarantee. Now, what databases, what source of molecules did we use? So really, the only two which were reliable and hand curated were RNAs-P database and 16S rRNA. Just to mention to those of you who are not familiar with that, RNAs-P cleaves off extra sequences of RNA on tRNA molecules, and 16S is in charge of binding tRNA, forming peptide bonds during the translation. So both are extremely important. One is about 16S RNA is about 1,500 nucleotide long, plus minus. There is lots of variance between species. RNAs-P is about 3 to 400. So this is slightly shorter, and this is typical typical secondary structure of both. Okay, so our results, our trees. So this is something that so this RNA's P3 that's based on 51 species and based on the secondary structure. So, okay, I mean, the coursing looks quite good. The bacteria, eukaryotes, and archaea are nicely separated. And if you look closely in bacteria, there's some coding there, BPH, BD, and so on and so forth. They also look reasonably good. I mean, I, of course, have no idea what these bacteria are, but if you look at this code, they're not, the ones which are close together are close in the tree, mostly. This is the same species, but the tree which is based on the sequences, not the structure, but just the sequences. So at least here we can see that the eukaryotes are not monophyletic anymore. So how do I do it? Like that. Okay, so we have the yeast here and the rest of the eukaryotes here. So at least in that respect, the structure is slightly better than the sequence-based tree. Okay, for 16S rRNA, we had just 20 species. Sorry, okay, I think these are 20 eukaryotes. They look quite good, except it's not quite clear. This group up here, it's not clear if it's monophyletic or not. The Drosophila and Amoeba, so I'm not sure exactly what they are, but the rest look at least reasonably good. This was the picture I had before. The 16S RNA from 91 species, not only eukaryotes, but eukaryotes, archaea, and bacteria. So as I said, it's a good coarse partition, and also with families within the bacteria at least. This color coding look correct. Okay, so after we completed this project, we found out, that somebody else did it, something similar. So David Penny's group in 2000, they were interested in something totally different. That's why we didn't find it initially. They were interested in comparing some catalytic RNAs, and among the things that they did a distance-based approach for R16S rRNA trees. This was sort of a side thing of their work. So we decided, but they used different. They just used the RNA distance package by Shapiro and Zhang. So we decided to take a close look at what their outcome was and what ours was. So the approach is really similar. Just the detail of what do you implement is a bit different. And they only cared about this thing, not other families. But so we decided to compare. Okay, one problem was that they had somehow 3 more structures than we had, and we weren't able. Wrote to David and told him 7 years ago what he wanted. They don't have the structures, so we could only use the ones that are in the database. So if we look at the archaea, there are 3 archaeas there. And it's very hard to look to see because it's sort of dark. So let's just look at the archaea thing, the archaea part of the tree. So we did it with just the 13 that were available, but the archaea still were in the same place. And I'll show it immediately. So the archaea are together, but they're arranged a bit differently. So in Collins, Davis group, we've got this Metanococcus as an outgroup. One is the secondary structure, one is the sequence-based. And in our case, we got the thermoplast as an outgroup. So, okay, what do you do with that? So in this case, we've got the structure. So we could try to take a look at the structure and see which one seems to be more correct. So we did that. So as you could see, there is this, okay, I can't say that these 2 guys which start with M, which in our case were together, and the thermoplasma was outside, and in Collins, one of them was an outgroup. So as you could see, if you look at the number of loops here, so this one has 1, 2, 3, 4, 5, 6, 7, and these guys have 2, 4, 5. So somehow, if this is a criteria, somehow this looks more like a little bit more outside than these 2. So if you just look at the secondary structure, and that's what the algorithm says, that then you would want it to be put outside. But that's not. I mean, probably just the algorithm that we use is probably less accurate. So that's not. So that's some difference, but not huge. Okay, so the result seems quite nice. Actually, we have some thought of extending that. Accuracy of structure-based tree seems comparable or slightly better than sequence-based tree. And finally, that's a warning. If you want to work on such data, you really have to get very good, hand-curated, or at least some human had to look at the structures. Otherwise, what you get will be very, very ridiculous. I could give you two very long sequences which just differ at one pair, and the algorithms produce, structures which are totally off and totally off the. These are structures with some function, totally off with the known function from the known function of this. So this is something one has to be careful about. And thank you very much.
So it's time for questions.
So the assessment you make of the accuracy of RNA structure-based trees and sequence-based trees is kind of qualitative in terms of.
Yeah. So basically, we looked if the eukaryotes are together or not together. We could have used So we had just had a talk about comparing trees. We could have used that, but it's not clear what you compare to. So
No, the question was if you try some quantitative measure, then it can guide you to fine-tune your assessment.
Okay, so
construction.
So I guess one thing is how different. Let's say I think one thing I want to compare is sequences versus structure. So I could say if the distance is not too large, then maybe it's good, maybe it's not good. But that's one thing. If not that, then I have to have some reference tree. So and which one would you take?
Okay. Well, I have my favorite. So, but
okay.
The standard is the NCBI taxonomy, but this taxonomy is not. It's a classification tree.
Yeah.
But anyway, you're using some gap penalties and particular parameter values. So this.
Yeah, that definitely I could maybe use that. Using some calibrating that against something that I believe or ready to believe, then definitely, as I say, the gap penalties and so on were fairly arbitrary. So this definitely could be something to tune things up.
The eukaryotic 16S RNA tree, was the eukaryote bit, is that based on mitochondria or mitochondria and chloroplasts? Because they ought to have two different origins within the bacteria.
How did Barbara? What was Barbara's method? I should send email to the students. I'm not sure. I've got the database here, so I could tell you what the database is, and maybe you could tell by that. But
the grouping together, presumably that's got something to do with the fact they've been isolated populations for 2 billion years, however long since they became endosymbionic, and so then have gone together.
So maybe it's worth looking. Checking, which is not too hard to do, but.
I was wondering, would it be a good idea to compare this method against just a traditional alignment method where you inform the alignment about the secondary structure? Like some programs allow you to do that, don't they? Like muscle and so on. You can give them sequences and secondary structures, and then they use that. And that would probably be pretty hard to beat.
Okay, we didn't do it, so but probably again it's a good idea. But I wasn't aware that you could that muscle is capable of doing it, but yes.
Okay, so we should thank all speakers again. So let's just say what the rest of the program is for today. So you've all got the piece of paper, but I'll read it to you anyway. Quarter 5, we have the welcome wine reception and then the poster session, and then and that goes until 6:30, and then that's it.
I don't know.
Yeah, you can.
Okay. It's okay, just portable thing. Okay. I will look here.
No, that counts. Yeah, humble.